Hi everyone, welcome back. This is the second part of our Meet the Clades Part 2. Part 1 covered lophophorates like brachiopods and bryozoans. This is going to cover the best and the brightest of the fossil record, the echinoderms. I am not biased at all. So looking at us right now are some beautifully weird and horrifying but still fascinating sea cucumbers who are feasting on a smorgasbord of small particles floating by in the ocean. So there you go. Super fun to watch, right? All right, so as we said, this is Meet the Critters Part 2. This is Sarah Sheffield lecturing for Geology 3104C at the University of South Florida. We pick up here the phylogeny of the Animalia. We've already talked about organisms without symmetry. That's our sponges, porifera. Our organisms with radial symmetry, which are the sister to bilaterians. That would be the comb jellies and the jellies. And we've already talked about some organisms within bilateria, the proterostomes, which covered bryozoans and brachiopods. Now we're going to flip over to here to the deuterostomes. We're not going to be following a really great order here, but let's continue with the echinoderms. They're defined by a five-ishness. Believe it or not, this is the real scientific term, a five-ishness. That means that echinoderms, as we saw on this tree, are bilaterally symmetrical. They're not pentaradial, like a lot of folks believe. And a lot of textbooks will actually tell you this, so please listen to me. All right? Textbooks are wrong. Echinoderms are bilaterally symmetrical with a five-ishness superimposed on top of it. That's what gives us this beautiful starfish shape, 1, 2, 3, 4, 5. This crinoid has arms in multiples of 5, as does this sea urchin has spines in multiples of 5. It has a high magnesium calcite skeleton made up of bazillions of little itty-bitty plates. The sea star has over 1 million plates in its body. That's why they make such beautiful fossils and such a disappointing few number of times that they are actually fossilized, right? And they have a water vascular skeleton. This means that the water vascular skeleton controls their movement, it controls their feeding, it can control respiration in a lot of cases. We assume that many in the fossil record had a water vascular skeleton because it is soft tissue, we really don't know for sure. Last note is that they are exclusively marine. They have no way to filter salt properly. So if they are in fresh water, they blow up like a balloon. This is obviously bad for them, and it does lead to their death. This was done in studies in the 1970s, from what I can only imagine were times before stringent ethics committees were formed. So do not ever throw starfish or any form of echinoderm into fresh water. It will kill them. So, fun fact, before we go to that horrifying slide, we have the sea stars, brittle stars, crinoids, sea urchins, and sea cucumbers. Now, there's a lot more diversity, but before we move on, I really want to focus on the sea cucumbers because they're mesmerizing and horrifying. We already saw them feeding, so let's watch what they do. This is obviously one pooing. This is a very important part. One of the things required to stay alive is to excrete waste, but also through their bum, they breathe. Let's watch this horrifying video. That is their bum, and they are doing what we call in the science world cloacal pumping. They have a cloaca tucked right up inside here with little respiratory veins running right on through, so they are actually breathing through their bum. This is not something that is, we're going to stop this video here while I'm talking. There we go. Now we can focus on words, right? Not this awesome bit of weird biology. So cloacal pumping is a method that many echinoderms likely did to breathe, especially maybe in lower or higher oxygen environments throughout the Paleozoic. We typically hypothesize it if an echinoderm didn't have noticeable respiratory structures and they had an unusually large paraproct, otherwise known as butt. The more you know, friends, right? Let's go to this next slide. Oh, nope, that restarted the video. Let's not do that. All right, so Echinodermata. This is a phylogenetic tree, there's a bazillion different iterations of this, and all of them are highly contentious with the 40 some odd echinoderm paleontologists in the world, yours truly being one of them. So what we see are these are the extant ones, meaning still living. 1, 2, 3, 4, 5, those 5 same groups. Crinoids are hanging out over here with a whole bunch of other dead stuff. we see a broad array of diversity, don't we? We have our blastoids, we have our crinoids, we have our rhombiferans, our parablastoids, crinoids, diploporans, cinctins, mitrates, eocrinoids, egioasteroids, all the living stuff, and helicoplacoids, which I assure you are echinoderms, even though they look like soft serve ice cream scoops, right? So we're going to talk a little bit about echinoderms and the fossil record today. Echinoderms have tube feet, like we said. They control feeding, locomotion, and they're controlled by the water vascular system itself. So here are the tube feet. Here is a good old friend, the starfish, crawling up the side of an aquarium. Areas that have these tube feet are called the ambulacral areas. This may or may not be the case in the fossil record. The ambulacral groove in fossil echinoderms may not have hosted tube feet. We don't know for sure. So this is more with modern echinoderms. So the water vascular system, how this works is obviously water is involved, pressure changes within the skeleton, move the tube feet itself, and that pressure is controlled by what's called the madreporite. In Greek or Latin, I can't remember off the top of my head right now, that is the stone canal. All right, so there is the madreporite on our good old friend the starfish. Here are tube feet, ambulacral grooves, and the pressure system runs along these rings here, which is how the starfish will move and do lots of things. Here's the madreporite on a chocolate chip starfish. That is its real name. My master's advisor in grad school had one named Amos. Get it? Ha. That is the anus right there. And this is the morphology. We have the top, where the madreporite again, the anus, and on the flip side, this is the mouth. Starfish can eat a shocking amount. So a lot of them can actually eject their stomachs through their mouths and swallow things like snails. It is horrifying and awesome. All right. Here are the ambulacral groove where the tube feet are. Right here at the end, there are actually, and you can see them here too. Do you see these little spiky things? Those are actually eye spots where it can see shadows moving. That's how it can hunt. Echinoids. So these are our sea biscuits and our sand dollars. You've seen these in lab already, so we're not going to go through these in terribly in-depth detail, but we'll go over it again. So as you remember from lab, in the regular echinoids, the nice little puffy sea biscuits, we have what's called a sideroid plating. Things are arranged in 20 nice, neat columns. We have the paraproct, the anus, sitting right on top. We have the peristome, or the mouth, sitting on the bottom. All right. Here are the ambulacral pores where the tube feet and the spines will attach. This is what they look like. Again, this is a really cute little one with lots of really nice plating. Gorgeous specimen. Inside the mouth would have been a jaw-like structure. This is called Aristotle's lantern. So they actually can move to chew things up. There are a number of evolutionary trends in echinoid echinoderms that we can talk about. So the big puffy round ones are the sea biscuits, and that's what we call the regular echinoderms, or regular echinoids, if you'll remember from lab. Irregulars are those that don't have that lovely symmetrical puffy shape, and these are the irregular echinoids. However, this is not a monophyletic grouping, and you'll remember from our evolution lecture, monophyletic means a single or a descendant and all of its ancestors. So this irregular shape has re-evolved. And the reason it's re-evolved is due to adaptive radiation. And this adaptive radiation deals with a more successful lifestyle. So the flatter test allows the echinoid to burrow under sediment a little bit better. Let's take a look at what that would look like. Here we have a heart urchin, otherwise known as a spatangoid, a fun science word right there. And this flatter test allows it to burrow a little bit better. And we can have deep burrowing echinoids, we can have shallow burrowing echinoids, but if you take a look here, the regular echinoids and the irregular echinoids, the regular echinoids are covered in spines that can serve a couple of purposes. One, this can help it stay a little bit more stable on soft sediment. And two, of course, it provides an excellent mode of protection, right? So if you saw one of those, you would not want to pick it up. Irregular echinoids don't have those large spines, and in fact they use their spines, which are quite small, almost they look kind of furry, to be honest, if you look at it without getting too close. These can help it move sediment out of the way. So it's a really useful lifestyle, and let's take a look at what that would look like. This is sped up by quite a bit, but you can see the little tube feet going to town, facilitating that burrowing. So this isn't going to be great for an escape mechanism because this is pretty darn slow, but it's really good for just staying under the surface, which can prevent a lot of predation in the first place. Awesome. So crinoids, this is another group that's still living today, extant. Crinoids today and crinoids in the Paleozoic are extremely different beasts, and the reason for that is so few species made it across the Permo-Triassic extinction that there's been so much genetic change that they're really not terribly comparable today, even though they are part of the same group. So sea lilies and feather stars, there's the same group, crinoids. Some have stalks, these little lilies. They look like flowers, but I assure you they are echinoderms, so animals. And then we have our feather stars, and both are just absolutely magnificent to look at. However, sea lilies, although they have a stalk, and many of us thought that they were fixed, and some are, we can actually see some of them crawling across the sea floor when they're threatened by predators. They can just pack up their literal bags and go. Pretty horrifying, right? But also pretty awesome. So crinoid morphology. I'm not going to ask that you memorize all of these. If we do work with them, I would expect you to remember where to find this information so that you can refer to them by the proper anatomical terms. But the big parts I want you to remember is that they have arms. These are the ambulacral grooves running down here. All right. The arms lead down here to the cup or the calyx where the mouth is, and the anal sac is around here in too, though that's rarely preserved. We have a number of different plates that make up the cup. You don't need to know their names, but you might remember these from the in-class exercise we did where we identified different plate types to test hypotheses of different species with radial plates, basal plates, and there's a third kind under here, the infrabasal plates, and many varieties of crinoids. And then finally we have our stem, which is made up of individual pieces. They look like Cheerios, to be honest, and that's a crinoid. Beautiful fossils. They have an excellent fossil record of disarticulated specimens. It's extremely rare to have them articulated because they are extremely delicate. Look at this beautiful fossil right here. All of these features are made up of hundreds to thousands of teeny tiny little calcite plates. But their stem pieces, as I said, they look like Cheerios. You can see that very clearly right here. Kind of looks like a big mash of cereal. Do fossilize quite well. Again, they're made out of high magnesium calcite, and they're quite abundant in marine sedimentary rocks, especially in the Paleozoic. This is an encrinite, what we call a limestone that's primarily made of crinoid pieces, and this is from the Mississippian, where the Mississippian is also known as the age of crinoids. So the Mississippian is in the Carboniferous, that is the time of the lower Carboniferous. There's a large group of extinct echinoderms called Blastozoans. This is a group that existed throughout the Paleozoic, and they all died during the Permo-Triassic extinction. These are a large group with hundreds to thousands of species, and each of the subgroups within it, there's multiple subgroups. So within Blastozoa, we have what's called blastoids, which is what you're looking at here. We have diploporans, which we'll talk about in a second, rhombiferans, things with diamond-shaped plates, and other groups, and each of them is identified by specialized respiratory structures. I'm not going to ask that you memorize any of these names, but I am going to ask that you listen to the concepts we're going to talk about. So each of these groups is defined by it. We originally considered these synapomorphies. Remember the defining feature of a group? Human synapomorphies are walking upright and unusually large brain. Blastoids have hydrospires. These are the respiratory structures that exist inside the specimen. So water will flow in through the top here. This is the oral surface here on A. It's up at the top. And water will flow in and circulate around these hydrospires, and then the deoxygenated water will come out another hole on top called the anospherical. That's how it breathes. Pretty cool, right? So here are a number of different early Echinoderms. Not all of these are Blastozoans, specifically D and F and K here, and J, excuse me. Those aren't Blastozoans, those are Asteroids, so kind of like a starfish on top of a cookie, if you can imagine with me. We find these a lot of the times attached to hard substrates like brachiopods. But we have our Crinoids here. Beautiful. Here's another Crinoid. These little holes, those are the respiratory structures on it. Here's an early rhombiferan. Beautiful fossils, the helicoplacoid. All of these lived in the early Paleozoic, and they have a really interesting pattern of evolution to them that can tell us a lot about how echinoderms evolve in response to climate change. Here is a diploporan, and the reason I'm showing you this is because we're about to look at diploporans themselves. This is the subgroup that I study. This is what my dissertation topic was on, and they're a pretty fascinating group, if I don't say so myself. So here's a blastozoan. You can see that five-fold symmetry right here, can't you? In the arm facets, 1, 2, 3, 4, 5. Beautiful, just like a starfish, right? The ambulacral grooves are here. These arm facets would lead to arms that come out or come off the top of the surface. We have the mouth, we have the butt, otherwise known as the anus or paraproct. We have the genital pores here where reproductive stuff would go on, and the rest of the body. Diploporans are identified by a special type of respiratory structure called diplopores, meaning double pores. How this works is there's a single depression, and in that depression on the plate of the body are two little holes. Presumably, these two holes acted as an in current and an X current with oxygenated water. So oxygenated water would interact with one hole, and then gas exchange would occur. Deoxygenated water would come out the other hole. That's what we presume. We're not entirely sure today. But as you can see, these fossils don't really look super duper similar to each other. It has a huge amount of diversity in kind of a small group. So here is what we think. My hypothesis is that diploporans are convergent, meaning diplopores are convergent. They re-evolve. And if diplopores themselves are convergent, we can't use them to define a group. You'll remember this from our evolution lecture, and the pterosaurs and birds both have wings, but that doesn't necessarily mean they're closely related. Here's an up-close picture of our double pore respiratory structure sitting within that single depression. This is a beautiful fossil, Haplospheronus from Estonia. Not all diplopores are constructed the same. As I said, there's a lot of broad morphology. Polycystis is from Indiana. Gorgeous specimen. This isn't a echinoderm, it is not an alien. And you can see that double pore respiratory structure, but this time it's connected by a whole bunch of different tubes. Now, I know I said that all diploporans have diplopores, but I actually lied to you. There's a couple that don't. Although we still consider them diplopores. This is Amphoricystis, and we have absolutely no idea how it managed to breathe. It is a mystery. And finally, recent fossil finds have identified completely separate echinoderms like Asteroids, the starfish on top of a cookie that I was talking about, has diplopores. Take a look up close. 1, 2 little holes. 1, 2 little holes. This is Thresherodiscus from Canada. So what this indicates is maybe we don't understand why respiratory structures evolved in the first place. Like what ecological trends or ecological factors are forcing these specific type of respiratory structures to re-evolve. Let's find out. So to do anything in evolution, we really need a phylogenetic tree. We made a phylogenetic tree. This here you're looking at is my dissertation topic. This is a phylogenetic tree of Diploporans with all the species names here. You don't need to know anything about this tree. Just identify that the subgroups of Diploporans are color-coded. The red ones grouped together, awesome. The yellow ones, not so awesome. There's a blue one out here. Green ones are actually non-Diploporans. So what we can see is that the Diploporans are polyphyletic. To jog your memory, polyphyletic means we have shoved a whole bunch of different organisms that are not closely related together because they look similar superficially. That means it's not an actual evolutionary grouping. So here we again have our little critters here, color-coded. The one group that is actually real, it looks like, are called the spheronodids, and they're identified by short ambulacral grooves and large arm facets. You can see those teeny weeny little ambulacral grooves and giant honkin' arm facets. Beautiful, right? Right. So fun fact, again, remember how I said remember how I said that evolutionary trees have a massive amount of solutions to them? This one tree with 29 species and 70 characters had 8.36 times 10 to the 26th solutions to them. Wild, right? So this does mean that it is likely that homoplasy is at play here with Diplopores. One potential hypothesis is that it was gained independently 3 times. The only way you'd know this is if you know the specimens themselves. The green ones primarily do not have Diplopores. The yellow and blue do. So maybe they were gained here twice, here again, and lost in Candida cystis, which is a non-Diploporan. Or maybe another way happened. Why would that be? Well, we're not really sure yet, but we think it has something to do with environment. Could it have been higher or lower oxygen? Maybe higher water velocity? We really don't know. Stay tuned. But the reason I bring this up is I wanted to give you another contextual example of evolutionary ideas. Here are your review questions. I will see you soon for your in-class assignment.